In this video, we will discuss about some of the key factors and signaling pathways that regulates ECM stiffness. So, first we will understand how the stiff matrices interacts with the cancer cells. So, they basically mainly interacts with these three receptors towards this ECM stiffness strengthening, that is integrin, beta 1, Piezol and PDGFRα. So once they interact with these three receptors, it will trigger downstream signaling pathways such as ROCK pathways activations, this RAS pathways activations, and this P13K EKT pathways activations, and this in turn results in the secretion of more collagens, and this secretion of more collagens in the tumor microenvironment will further leads to the strengthening or contractions. This in return leads to the increase in the stiffness of the ECM. So how this cellular matrix collagen stiffness increases? It's mainly because of the enzymes that comes from the family of LOX, and this LOX is a collection of enzymes that comes from PLOD, and it's a copper dependent enzyme. And the function of LOX is that they behave differently in the stiff environment and they behave differently in the soft environment. So this LOX has usually does is hydroxylates lysine into the high aldehyde-derived collagen, or in short people call it as HLCC. So once this hydroxylates occurs, then it will change the matrix compositions, and in the stiff region, in the stiff environment, the HLCC is more dominant and the LCC are not, whereas and some of the examples of the stiff tissues are near bones, whereas in the case of where there is soft matrices are present, LCC is more dominant. LCC is lysine aldehyde-derived collagen, and those are more common in the soft tissues such as connective tissues. So this LOX plays an important role in that, and others also plays a role, but the key roles here that is shown in this figure is about this LOX and the MMPs. MMP's role is to cleave the cellular matrix, thereby helps the proliferations or migrations of these tumor cells. And this other important pathways is this HIF-1 alpha. It is basically to decrease the supply of oxygen or create a oxygen deprived conditions, which will help the cancer cells to proliferate. And this there is also mention about this ECM stiffness also depends on this, as it is shown here, Piezol and integrin beta 1. Once it is stiffened, then it will interact with this integrin beta 1 and Piezol, and this in return will leads to the further strengthening of this ECM stiffness, because it will release more collagen. And also this tumor microenvironment will also have more amount of lysine, because the lysines are secreted by the cancer cells, and this lysines, once it is present in more amount, what this LOX does is it will hydroxylates those lysine to make HLCC, that is high aldehyde-derived collagen, and this will lead to the further strengthening of this ECM. So this is how the cascade cycle works. I hope you understood in brief the cascade cycles, that once this Piezol and integrin beta 1 activates, it will trigger the release of more collagens, and the cancer cells will also goes under cellular changes in such a way that it will release more lysine. And once the surrounding environment of the cancer cells has more lysine, and this LOX, it will convert those hydroxylates, those lysine, to make high aldehyde-derived collagen or HLCC, and this will lead to the strengthening of the CCM. So I hope you understood now when I have explained this cycle like 2 times in this video. If you have still doubt, needs to go through this whole pathways or factors or signaling pathways that are involved in CCM stiffness. You can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time. And I have not discussed all the points individually. I have also only covered those that are important. If you want to know about all other pathways, you can refer to the reference. Thank you again for your time.